Illustrate the life cycles of Ectocarpus with suitable example. The life cycles of the brown alga. Ectocarpus is a classic example of isomorphic alterations of generations, where a difold sporophyte phase alternates with the haploid sporophytes phases. Both generations are morphologically insimilars. The most studied example, Ectocarpus is siliculosus. The cycles involve both asexual and sexual reproduction methods. The dipole sporophytes, the dipole sporophyte to N. The dipole sporophyte to N. The life cycles begins with the haploid sporophytic plants, which is a sexual generations, which is a sexual generation. These plants involves the two types of spongia. This plants produces two types of spongia. Unilocular spongia. These are single-celled organism. These are single-celled organism in large terminal structure on the branch. Terminal structures on the branchlets. Inside the dipole nucleus undergoes meiosis. Undergoes meiosis. इसके बाद फिर क्या undergoes meiosis. Undergoes meiosis followed by mitotic divisions to produce haploid 32 से 64 haploid N nuclei. Each nucleus develops into biflagellates. Each nucleus develops into biflagellates. Each nucleus, the zoospores are released. The zoospores are released. इसके बाद फिर क्या बोलते हैं इसे, zoospores are released and each and a substratum, the cells in substratum. Zoospores are substractums. Germinate to grow into the haploid gametophyte plants. इसके बाद फिर क्या बोलते हैं, फ्लोरोकुलर स्पोंजिया. These are multicellular concept structure and formed by the mitotic divisions. Formed by mitotic divisions. On terminal cells. Formed by mitotic division on terminal cells. Cells within the gametosium produce haploid biflagellate zoospores and the mesospores. The release are means of rapid asexual reproductions. They grow into a new haploid sporophytic plants. They grow into a new haploid sporophytic plants, effectively cloning the parent generations. The haploid gametophyte plants. The haploid mesospores from the unilocular spongia. The haploid gametophyte plants, which are the sexual generations. इसके बाद फिर क्या बोलते हैं, फ्लोरोकुलर स्पोंजिया. The gametophyte plants are the gametophyte plants and produces on the concept structure called floricular spongia, which are morphologically similar to the floricular spongia of the sporophyte. इसके बाद फिर gamete formations and fusions. The cells within the gametophyte produces biflagellates and by flagellates gametes via meiosis in a species like Aeschylocoeles. Mm, female gametes are morphologically identical, isogamous were physiologically different, anisogamy, and the male gametes are released male gametes are released male gametes are released male gametes are secreted by the senescence, are the female male secreted the female male are the female male secreted gametes, gametes नाले मेल secreted gametes. इसके बाद leading to a leading leading to a leading to leading to for leading to a clump of formations and fusions and zygote formations नाले the fusions of a male and females. Gamete diverges on the two end diverged zygote. इसके बाद फिर क्या बोलते हैं से टू एंड जाइगोट डाइवोल्ट जाइगोट नहले. इसके बाद फिर क्या बोलते हैं से अथी नहले बताते हैं कैसे जाइगोट फॉर्मेशंस हो गए. एक बाद फिर क्या बोलते हैं से अथी नहले जाइगोट फॉर्मेशंस. इसके बाद फिर क्या बोलते हैं से डेवलपमेंट्स नहले the zygote does not develop in immediate meiosis into involved germinated mitotically to develop directly into a diploid sporophyte plant completing the life cycles. इसके बाद फिर completing the life cycle ना फिर क्या बोलते हैं pathogenesis. Occasionally in the absence of the compatible mate, a haploid plants develops into a new plants without fusion pathogenesis. इसके बाद फिर क्या बोलते हैं sufficient parthenogenesis नहले. And these plants can be asexually reproducing parthenocytes and gametophytes depending on the species and environmental conditions. Example Ectocarpus aeschylocoeles. Ectocarpus is the primary model organism for studying the brown algae. Due to its manageable and rapid life, cycles completing in about 3 months in a lapse, and the clear demonstrations isomorphic alterations of generation. The specific illustrates how to dipole and haploid and dipoles. Phases are distinct, non-living, and morphologically similar. Involves in nature. Answer complete.